Okay, so this is my video number 2 in my genetics unit series. So we're going to look at Punnett squares now. So let's go ahead and go back to that family that I started my last video about phenotype and genotype with. So let's pretend that these two parents both have the brown eye phenotype. So they have brown eyes as their phenotype, but their genotypes are that they're both heterozygous. Now, what are the possible genotypes and phenotypes for their children? So by the end of this video, you'll be able to answer simple genetics questions like this. So let's go ahead though and look at kind of connect genetics to meiosis a little bit and see what's really happening when we inherit our traits from our parents. So here, this person on the left, like we have 3 different possibilities when we talk about genotypes. So a person could be homozygous dominant, heterozygous, or homozygous recessive. Those are going to be like our 3 options when we do genetic word problems that are for autosomal chromosomes, and we're not talking about sex linked with X's and Y's. So now let's go ahead and look at the father of our family. So he's heterozygous, and you can see that means that he has one dominant allele, like brown eyes, and one recessive allele for blue eyes. So he has 2 different alleles in his genotype, but his phenotype is brown eyes. So this is a diploid. We are diploid organisms. Here he has 2 copies, but he only passes one to his offspring. You get one from your mom and one from your dad. So somehow he needs to divide his 2 copies so that only one is passed to the offspring, and that's where meiosis comes into play. Like that's the whole purpose of meiosis, is so that each of your offsprings inherit only one of each kind of gene. So anyway, so as we go through meiosis, these chromosomes are going to separate. So at the end of meiosis, you end up with 4 gametes. Now, when we look at this man, this individual, he is heterozygous. So what we noticed throughout the process of meiosis, his alleles, his dominant allele and his recessive allele, they separated, right? So here, this represents the possible gametes that this individual that he can make. So there's a 50% chance that he'll pass that dominant allele onto his offspring and code for brown eyes. And then there's a 50% chance that he'll pass the blue eye allele onto his offspring. Because when meiosis occurs, these diploid organisms, the dominant and the recessive in this case, the 2 alleles, they separate. So half his gametes will have a dominant allele and half his gametes will have a recessive allele. There's a 50-50 chance that his offspring will inherit the dominant and a 50% chance it'll inherit the recessive. So let's just look at a different example, not dealing with that guy. So here, for example, let's kind of see how setting up Punnett squares works very simply. So here, this father on the left is homozygous dominant. So he has 2 brown eye alleles. And then, so when meiosis happens and they separate, there's a 100% chance he's going to pass brown eye directions to his offspring. Like that's all he can pass on. 100% of his alleles say brown eyes. Now, if we look at the mother, though, as she goes through meiosis to make eggs, she only has blue eye directions. Both of her genes are recessive. Both of her alleles say, here, let's make blue eyes. So if here we use a Punnett square to see, well, what kind of children could they make? What would their children's phenotype and genotype be? So in Punnett squares, what we do on the outsides of the Punnett squares, that's where we write what are the possible gametes from the parents? Like what could the parents contribute or give to the offspring? So a Punnett square is just showing what are the different possibilities that could happen in offspring between 2 parents. So here, in this box of the Punnett square, it's like saying if this egg from the mother was fertilized by this sperm from the father, the offspring would be heterozygous and have brown eyes. And that's true for all of these, right? Like that's all the mom can give and that's all the dad can give. So 100% of their offspring would be heterozygous and have brown eyes. So let's go back to our family we started with. So let's see, what are the possible phenotypes of their children? So here, when we look at the Punnett square to see the possibilities of kids they can have, we see that the mother, her gametes, when they're made by meiosis, are going to have only one allele each. So her gamete or her alleles separate during meiosis, and so there's a 50% chance she'll pass on a dominant allele and a 50% chance she'll pass on a recessive allele. And the same with the father, who's also heterozygous. There's a 50-50 chance. So when we use a Punnett square, what we're going to do is we're just seeing what are the chances, like what are possible in offspring from these two parents. So the first offspring could inherit a dominant allele from both parents. The second possibility could be a dominant allele from the mother, but a recessive allele from the father. A third possibility could be a dominant allele from the father, but a recessive allele from the mother. And a fourth possibility could be a recessive allele from the father and a recessive allele from the mother. So we look at, the question was, what are the possible phenotypes of their children? Well, this child will have brown eyes because of that dominant allele. This child will have brown eyes because of that dominant allele. This child will have brown eyes because of that dominant allele, and this child will have blue eyes. But really, this family isn't going to have 4 children necessarily. The Punnett square, the 4 boxes, are showing us the likelihood or probability of these phenotypes happening. So it's only showing us the likelihood that something will happen. So in reality, if I look at all the possible combinations between these two parents, there is a 75% chance that they'll have a child with brown eyes. There's a 25% chance their child will have blue eyes. So their very first kid could have blue eyes. It's just a 25% chance. Their second kid could have blue eyes, but there's just a 25% chance. So there's more likelihood it'll have brown eyes, but could also have blue. Okay, so now though, let's go ahead and see what are the possible genotypes of their children. All right. So in the first one here, there's a 25% chance they'll have a homozygous dominant offspring. There's a 50% chance they'll have a heterozygote offspring. And there's a 25% chance they'll have a homozygous recessive offspring. Okay, so now what we're going to do is I just have 3 examples to help us practice with setting up Punnett squares. So here, and I made all 3 of these up, okay? So in roses, having thorns is dominant to no thorns. So one of the most important things to do when reading word problems for Punnett squares is you always want to start with, like, no matter how long the word problem is, whenever you read what is the dominant trait and the recessive trait, you need to pause and write it down. So here, this problem is dealing with thorns, so I'm going to use a capital T. So here says having thorns is dominant. So my capital T represents thorns. Now my recessive trait in this flower, or the recessive trait, is no thorns. So now let's go ahead and see the problem. It says if a heterozygous plant is crossed, crossed means they're mated. Like you're going to take a sperm or an egg from one and a sperm and an egg from another, or an egg from another, and you're going to mate them. So crossing means like taking gametes from each and like fertilizing them to have offspring. Okay, so if a heterozygous plant is crossed with a homozygous recessive plant, what are their possible phenotypes of their offspring? Well, here are our 2 genotypes. So because they're flowers, it's not really like males and females in roses. We'll just say like the mom, let's say she's heterozygous. So that means she's going to have a capital T and a recessive T. One of her alleles says have thorns. One of her alleles says no thorns. And then the other parent is homozygous recessive, meaning it has 2 of the same homozygous, but it's recessive. So the dad would have no thorns because that would be his phenotype. Okay, so now let's do a Punnett square for these 2 like parent plants. So here, when we look at how you fill it in, you take I don't like that. Okay. So now our question was, what are the possible phenotypes of their offspring? So we're seeing, well, what do they look like? Well, I remember from what I wrote down is that the dominant allele is thorns, and I can see two out of their four offspring, 50% of their offspring will actually inherit that dominant allele. And you only need one dominant allele to actually have that dominant phenotype. So here, about 50% of their offspring will have thorns, and about 50% will have no thorns. So those are the phenotypes that are possible from these two parents. Now, let's go ahead, though, and look at another example in humans. So straight thumbs would be like this, and then this one's better. Curved thumbs would be like that. So straight thumbs is dominant to curved thumbs. And I think that's true, dominant recessive. Okay, so obviously I'm recessive. So now, again, the first thing you want to do is write down what is dominant and what is recessive. Again, I'm using thumbs, so I'm going to use a T. Now, sometimes you could pick the dominant trait as your letter, so like straight thumbs to curved, but an S or a C, those aren't very good letters to use because if you're doing your Punnett square, a capital S looks very similar to a lowercase s. So then later, when you're like looking for results, it's very easy to get mixed up. Same thing with a capital C and a lowercase c. So always try to use letters that look different, like a B or an E or a G or an R, where there's like an obvious difference between your capital and your lowercase. So that's why I chose a T here for thumbs. Anyway, so a straight thumb is dominant and a curved thumb is recessive. So our problem says, if a true breeding straight thumb individual is crossed with a curved thumb individual, what are the possible genotypes and phenotypes of their offspring? Okay, so true breeding is another way of saying homozygous. So I have a true breeding straight thumb. And if I look at the left side of the screen, a straight thumb is dominant. So I know one parent has a straight thumb, so they have at least one capital T. But then it says true breeding, which means like purebred or homozygous. So I know one parent is homozygous dominant for straight thumbs. Then it says a curved thumb individual. Well, the only way you could have a curved thumb, because it's a recessive trait, is if you had two copies of that recessive allele. Because if you were heterozygous and had a big T and a small t, you'd have a straight thumb. So the only way to have a curved thumb is to be homozygous recessive. So when we fill in this Punnett square, here, we can see the possible genotypes and phenotypes. 100% of their offspring will be heterozygous genotype. And 100% will have straight thumbs. It is impossible for this couple to have any curved thumb children because the mom, the female here, she only has dominant alleles to give. That means in meiosis, 100% of her eggs will always have that dominant allele. So 100% of her offspring will have straight thumbs. Okay, so let's go on to my last example. Here it says, in grasshoppers, short antennae are recessive. Okay, so sometimes you're going to get tricky questions where instead of directly telling you what is the dominant trait, they'll tell you what the recessive trait is, and then you got to like work backwards. So here, recessive is short antenna. And then that would mean that long antenna is dominant. So if two hybrids, a hybrid is another way of saying heterozygous, or heterozygotes. So if two hybrids are crossed, what is the likelihood that they'll have a short antenna offspring? So here I have one heterozygote parent and another heterozygote parent. Now, as I fill in my Punnett square, what I notice is this first parent is going to have a long antenna phenotype because it has two dominant alleles. Now, let's look at this next one. You notice that I put the capital A first and then the lowercase a on the right-hand side. When you have a heterozygote, you always write the capital letter in the front, kind of like when you write your name, you write it with a capital letter to begin. Okay, so this offspring, though, is going to have a long antenna phenotype because it has one dominant allele. And then this third offspring would also, or I should say the third possibility in offspring, would also have long antenna. And it's not till this fourth combination where you have two recessive alleles come together in the zygote that you get a short antenna offspring. So really, the likelihood of these two grasshoppers having an offspring with short antenna is 25%. So it's only a 25% likelihood they'll have a short antenna offspring, but a 75% likelihood that they'll have offspring with long antenna. All right. So those are my 3 examples, and good job.